and then turn my.
Just let me know. Just let me know. Okay, I have it as live. Welcome back to the Early Career Immunology Seminar Series. For those of you that are returning, thank you for your continued support. If this is your first time in this space, allow me to introduce myself. My name is Tim O'Sullivan. I'm an assistant professor of immunology at UCLA. The purpose of this seminar series is to increase the exposure of early career faculty in a time which we haven't achieved equity and representation at scientific conferences and seminars despite equally innovative discoveries. We've had several speakers speak already, and you can see that some of their work has already been published in high impact journals. And so if you sit on an organizing committee for seminars or for conferences, think about inviting some of our speakers. And I know that they'll greatly appreciate it and provide you high quality content. So today we're excited to have Justin Perry with us. Justin is the technical mastermind behind our seminar, and we're all thankful for his tireless efforts in providing you all quality streams. Justin focused his PhD studies at WashU, and he focused his PhD work on trying to understand T-cell tolerance development. Specifically, he studied the contribution of apoptotic cell clearance by thymic antigen presenting cells in their contribution to T-cell tolerance. During his postdoctoral training at UVA with Cody Ravichandran, Justin explored unanswered basic fundamental questions on apoptotic cell clearance. Now with Sloan Kettering Institute's immunology program, Justin's lab seeks to understand the role of these aforementioned programs used by phagocytes during homeostasis and when homeostasis is perturbed. And as would be expected of any newly hired MSK PI, Justin has quickly racked up multiple early career awards, including a V Foundation scholarship and recently a Pew Trust scholarship. So this is in the chat, but I'll mention this again. If you have any questions for Justin during his talk, go ahead and type them into the chat box, and I will ask these questions to Justin at the end of the talk. Justin, thanks for being here today, and we're all excited about the work that you'll present.
Great. All right. Classic share moment. All right. Okay. Yeah, so I'm Justin. Our lab studies phagocytosis and broadly speaking, innate immunity, although as you will come to see, innate immune cells aren't the only cells capable of performing this process. So in the human body and generally conserved across all species, we have evolved several ways to die, for cells to die. And in fact, it turns out that death is necessary for life to exist. In fact, most tissues turn over at high rates. And in fact, a recent paper suggested that there's approximately 3 million cells per second are cleared from the human body. It's approximately 3 billion per day, or between 3 and 30 billion per day. So it's about 1% of your total body mass that's cleared every day. And most of this is cleared through regulated cell death, particularly a form of cell death called apoptosis. But as you can see, there are actually several ways that we have evolved to do this cell death process. And so we will be focusing on particularly these apoptosis, and more importantly, not just how these cells die, but also how they are cleared. And so my lab focuses on the clearance of these cells, a process known as efferocytosis, or literally eating of self. And so in this case, efferocytosis is highly conserved. For instance, we see this in the larval development stage of C. elegans, or roundworm species. We see this, for instance, in the optic tectum of the developing zebrafish. We'll see it in the midline of the developing fruit fly, or as we know them as Drosophila. In one of my favorite species, the Hydra, or Cnidaria, is not only necessary for wound healing, but also for full regeneration. So if you cut a hydra in half, you can see pretty clearly when it grows back together, there's this wave of cell death and cell clearance. And this is also true in my absolute favorite species, the axolotl or the Mexican salamander, where if you cleave a limb, you can see that this limb will fully regenerate, and that depends on both cell death and clearance. And the clearance of these dead cells is necessary to both clear up space for proliferative stem cells to repopulate and regenerate the tissue. But it's also necessary to secrete factors that contribute to this process. And of course, we see this in mammals as well. For instance, in mice, you can see here that the developing mouse has waves of cell death in the webs in between fingers. And in fact, this is true in humans as well. So if you fail to clear during development, this is a common a rare but seen complication in human development, where you can see that you don't separate the webbing in between the fingers, and you have dactyly formation in this case, or the fusing of fingers. So it turns out that efferocytosis is actually essential for organismal homeostasis, broadly speaking. So pretty much all major tissues and organs have dead cell turnover and clearance of those dead cells. As I noted before, it's billions and billions of cells, the vast majority of which we know are red blood cells, neutrophils. These turn over each day. There's replenishment of these cells, and this is often linked to circadian clock, for instance. And every tissue or organ also has multiple different phagocyte types, or cells that are capable of clearing these dead cells. So just as a classic example, in the lung, we have in the airways, you have alveolar macrophages that are necessary for cleaning dying epithelial or shed epithelial cells, and as well as the surfactant that is produced and used up. But in the same airways, you also have epithelial cells that are capable of clearing their dying neighbors, or at least particulate or parts of their dying neighbor. And it turns out that if you perturb this, at least genetically, or in for instance during aging, each one of the perturbation of this process in each one of these tissues results in pretty catastrophic consequences to the host. Of course, the conventional two forms being atherosclerosis, or plaque formation in the arteries, or in systemic lupus erythematosus, which is the canonical cell clearance disease. So my lab studies tries to understand how that is that a phagocyte can clear dead cells. Because if you think about it, when a cell dies, it doesn't just release all of its contents. In fact, the cell packages it into these tight cargoes, if you will. And these cargoes contain lipids, amino acids, nucleotides, and other sorts of debris and material that a phagocyte that clears that has to deal with. So if you really think of it, there's all of this biological material, all this biological stuff that a phagocyte has to deal with. And so in order to do this, there's a series of steps that have been really well defined by the Ravichandran lab, by the Nagata lab, and many others at this point. So initially you have the death of a cell that releases recruit me signals. Also, we colloquially call smell signals. Historically, this has been known to be ATP or other nucleotide phosphates that have been released that we knew would recruit phagocytes. But more recently, the Ravichandran lab and others have shown that actually turns out apoptotic cells also release other metabolites and other interesting secreted factors that communicate with the phagocyte. However, once the phagocyte gets there, it still has to distinguish between the live cell and the dead cell. And so this is done through canonical signals that are exposed in the dying cell called phosphatidylserine. So it's essentially the exposure of the inner leaflet of the plasma membrane is exposed out. And that's recognized by a panoply of receptors at this point that are extremely redundant in every cell type. And that very triggering is actually known to already secrete and release, produce various wound healing factors that will recruit other cells to the neighborhood, these sorts of things. That PS engagement with the receptor also triggers actin polymerization, triggering the next step, which is the physical encapsulation or internalization of the corpse. So we call this the engulfment or internalization step. Once that cargo is brought in, you will have a series of lysosomes come and fuse to that phagosome. And this is where you get the processing or degradation step of clearance. And collectively, we term these ingestion signals. So it's these latter two signals that have been less well studied over the years. To us are probably the most interesting ones, as you will come to see later, and has been the main focus, particularly the metabolic regulation of these steps, as well as as you'll see in the story I'll tell you today, the ion regulation of these steps. So what does this actually look like? And so I happen to subscribe to the idea that a picture or a video is worth 1,000 words. So what you see here is, excuse me, is a macrophage labeled in green, engulfing multiple corpses that are labeled with a pH-sensitive dye called Cypher 5e. This dye reacts to a decrease or acidification of the lysosome, so pH change, and turns pink. And so as you can see, actually we can learn a lot of things from this video, but you see that when it takes up that corpse, you see that it transitions into the acidification and then begins to break that corpse apart. And then you see another corpse taken in, lysosomes fuse, and then it starts to disassemble that corpse. So this is those latter two steps of internalization and digestion and the eventual digestion of the corpse. So about 2 years ago, time is a flat circle at this point, as everyone who's also dealing with COVID knows, became really interested in this idea that maybe we could, in a big data way, tease out some of these signals that are induced after the internalization of the corpse. And so we used a sledgehammer trick where you can basically block the internalization and digestion of a corpse using cytoclasin D, which prevents actin polymerization, basically allows for the external signals, preventing the internalization. So we did that, and then at the time, we didn't have many good ways of resolving the material from the corpse and the phagocyte. And so we used, for instance, the mRNA that is quite stable in a lysosome. So at the time we used a mixed species approach where we used a hamster fibroblast, which is an extremely phagocytic cell, and fed them human Jurkat lymphoma T cells, apoptotic Jurkat lymphoma T cells, and then performed RNA sequencing. And in fact, there were a host of signals that were induced after the internalization of the corpse. But almost disappointingly, there weren't a lot of patterns that were unique, at least on a program level or a gene program level, that were different between contact or soluble factors and the internalization factors. Really, the only thing that was uniquely different was this program that was upregulated after internalization that involved chloride transport and cell volume regulation. Now, ever the optimist I was, we started thinking, okay, well, this is an interesting question because we know that phagocytes, when they eat a dead cell, they don't explode. They take up all of this biomass. They handle all of these unique metabolites and ions and other stuff, water and other things. And they really don't change a lot in size. This is a known phenomenon for decades that no one really ever knew why or how. So to make a long story short, one of the pathways that we found was this chloride sensing signaling pathway, basically that starts with chloride sensing directly by these kinases with no lysine 1 or Wink1 that basically directly binds chloride. And so in the presence of chloride, this pathway is inactive. And then when chloride is relatively less or leaves the cell or there's less chloride available, that auto activates Wink1, which then in turn phosphorylate themselves and subsequent downstream effector kinases, SPAC and OSR1. And then those kinases simultaneously phosphorylate these two transporters. One is a chloride importer, the other is a chloride exporter. And so the chloride And so this basically it stops chloride efflux from the cytosol and allows the chloride to come back in. And so what we found was that if we perturb any of the components of the influx pathway, so basically prevent the ability to influx chloride back into the cell, it turned out that the cells actually overate. So they ate way more corpses on a per phagocyte basis. So they were overeating. This was pretty remarkable because this was a classic loss of function assay that actually resulted in a gain of function. Whereas if we inhibited or knocked down or deleted the other transporter, they actually failed to eat, or they would eat much less on a per cell basis. So pardon my dad joke, but immediately I thought, is this overeating unhealthy, or is this healthy eating? And naturally my children, who are not much older, would disagree with me on whether it's healthy or unhealthy to overeat potentially dangerous or noxious material. Actually, we might agree on this point that overeating candy is okay. It's fine. And so we turned back to our mixed species approach, but this time we sorted the active engulfers. So you can see that they're eating much more. We sorted the overeaters and did another round of RNA-seq. And so what you would normally see in control active engulfing cells is this distinct program, basically a general suppression of pro-inflammatory program, an activation of an anti-inflammatory program, and a suppression of an oxidative stress program. And we really saw the reversal. Either they failed to induce anti-inflammatory program, or they began to induce a pro-inflammatory program, and they seem to be under much more oxidative stress. And importantly, I think it wasn't just because they were overeating. It was specifically the absence of this transporter, because if you overexpress the phosphatidylserine receptor, TIM4, so this means that these cells can eat more because they have more receptor on their surface, they eat more cells. It didn't seem to induce the same inflammatory program, at least as assayed by the most differentially changed genes that we tested. And so it seemed to be very specific to this knockout. And so at the time we claimed that this pathway was necessary for interpreting an engulfed apoptotic cell as anti-inflammatory. So the astute observers in the audience will already be wondering, okay, so you're telling me that you have to efflux chloride in order to appropriately engulf a dead cell. But if you do that, that makes the cell more pro-inflammatory, right? So you're basically telling me that you efflux chloride, you eat more, that puts you in an immunogenic state, or just that's a fancy way of saying that these cells are cranky, and that you have to then influx the chloride back in to both stop eating and switch to a tolerogenic state. And if you don't do that, then you're back in having a cranky cell again. So this seems really counterintuitive. And in fact, it is very counterintuitive. It's not what you would think, because historically anyway, in our field, the model has been that efferocytosis directly induces a tolerogenic response. I showed you this from the beginning. Simply just touching a dead cell can induce release of IL-10 and TGF-beta, for instance. And so the model has been that if you eat, you induce a tolerogenic response, and then if there's no danger sense, no pathogen, no, nothing like that, then you maintain tolerance. But if there is a pathogen sense, there's some sort of danger, DAMP or PAMP sense, then you would switch to an immunogenic state. But what our data was suggesting, and this is what we are at least what the lab proposes now, is that in fact, efferocytosis is inherently dangerous. And so when you eat a dead cell, it primes for an immunogenic response. If there's no danger sensed, it would switch to a tolerogenic state. So you're good, the cell's fine, there's no danger, imminent danger, the neighborhood's fine. But if there is a danger, you're already primed for that danger response, and then you can immediately switch to an immunogenic state. So of course, if that were true, let's say, then you would need a switch that's extremely rapid to go from a primed to be immunogenic state to a tolerogenic state. So I know this is a lot of that sort of circuit logic, and so I think it really helps to illustrate how this could play out, at least how our hypothesis is that essentially what we think, and we think that the Wnt1 SLC12 circuit is in fact one of these rapid response circuits, essentially where if you eat a dead cell, that induces this state change. Basically, you're primed and ready. And then if the phagocyte senses that change, it switches on this circuit. And then if there's no pathogen, it has to then revert back to homeostasis and basically close up shop and revert back. And so that's why you would have chloride influx via SLC12A2. If the levels are unsafe, just during homeostatic clearance, or if there's a pathogen, the circuit remains open, and then you have an immunogenic response, so an antibacterial response of some sort. And actually, it turns out that a paper just came out recently that this pathway, the Wnt1 pathway, is actually necessary to suppress the inflammasome response. So NLRP3 inflammasome, this is in fact the case. So this was a bit of a scoop for us as far as that goes, and we'll show more data on the homeostatic side in a minute. But I do think it's worth mentioning and giving credit to that this did just come out. That in fact, it does seem that this is in fact at least a circuit for regulating the inflammasome response. So we wanted to test this circuit. This was kind of the goal. And so I'm going to present the story now from two postdocs teaming up, Julia and Alyssa. So we wanted to test if this was true. I mean, first of all, is this ion flux rapid? We had never really studied chloride flux in phagocytosis. We wanted to see if this inflammasome aspect, we were already working on the inflammasome side before, and we wanted to see if there's any in vivo impact of this pathway. And so we'll kind of go through those bit by bit. So in order to do ion, in this case, chloride imaging, we use this really cool chemical called MQAE. MQAE is really useful because most indicators, and we don't often pay attention to this, but this actually does really matter. Most indicators are sensitive to levels of sodium bicarbonate, of the pH in the cell, and all sorts of other factors. And in fact, that can change their brightness simply by changing the ion concentrations inside of a cell. This one is not sensitive to all of those. This one does not localize to any organelle or lysosome inherently. It can as part of a bigger process, but it won't just in a steady state cell. And it's actually seems to be a pretty imageable marker. And so the way that works is that you label a cell. If the MQA is quenched by chloride, you'll have a dimmer signal or black space as far as microscopy goes. And if there's not quenched, so if there's chloride efflux, for instance, you'll have a bright signal. So it takes a moment to switch the brain over to thinking kind of in the opposite way, where bright signal means less stuff and dim signal means more stuff. But let's look at some imaging of this. So here in these experiments, these are control wild type macrophage performing phagocytosis. Green here is MQAE label. You can already see that there's apparently a dearth of steady state chloride in the nucleus, which is interesting in and of itself. In this case, you have lysotracker labeling the lysosomes in the phagocytes, in the macrophages, and then you have our favorite pH-sensitive dye labeling dead cells. So let's go ahead and look at a few of these. So here you can see this is pretty rapid imaging. You can see that this macrophage is in the plane of view. You can see it eating both. You can see the macropinocytosis occurring that just normally occurs. Then you can also see there is clearance, dead cell clearance occurring. And one of the things I want to point out is that you can see these rings and really clear regional flux of chloride. So let's look at another one to show you. There's another cell. You can see that there are these pulsing waves of chloride flux. It seems to be fairly regional. I'll give you one more example. This one is my favorite. So you can see when it brings in 2 corpses on each side before breaking them down. This one, and you can see these beautiful rings of chloride flux as it fuses with the phagolysosome. And so this is what this would look like here. You can see that, again, in my favorites, if you watch just the pink for a minute, you get the ring in, and then it acidifies, boom, right here. And then if you watch the lysotracker, you can see that the lysosome comes in, and then you get this beautiful, beautiful acidification. And then this chloride flux is extremely rapid. And then we can actually quantify this. So I just wanted to show you that Julia came up with a nice way of inverting and being able to quantify this, these changes in these waves of chloride flux. So what about the knockouts? What do these look like? So you will already tell several observations, and this is why I love microscopy, because you can really observe lots of unique things. So here you have a knockout cell, and you can already tell their chloride is disrupted. They don't have these really beautiful pulsing waves of or rings of chloride flux around, despite the fact that they're still eating quite a lot. And this is another example. So you see here, it just took one up. Really no, I mean, that was a massive corpse uptake, right? And there's really no change in the chloride flux, at least as we saw in the controls. And one more example, again, you know, and there's clearly a pretty big difference in their chloride flexibility. And so if you look, this is what this looks like in that real time. You know, clearly there's, remember, bright means less chloride. So clearly in the concentrated core of these phagolysosomes, there's a dearth of chloride. And that's probably because chloride has been effluxed from the phagolysosome into the cytosol and out of the cell, but it's not bringing it back in. So there's really probably a unidirectional flux at best. And again, that's what this looks like. So we can very clearly quantify this and look at these sorts of waves. And so what Julia found was that there seems to be distinct pulsing waves of chloride. First, when you find and taste the corpse, you see this really high chloride burst, and then it rapidly decreases when you ingest the cell. And you saw those were those chloride rings surrounding the phagolysosome. And then during the digestion, then you basically have the lowest period of chloride in the phagolysosome. And that's basically the gray bars here. This is an accumulation of hundreds, if not thousands, of cells analyzed. And you can see, you know, basically you have these beautiful peaks. Here you have your highest loss of chloride, as I mentioned here. So basically the biggest efflux of chloride during ingestion, and then it rapidly goes the other direction during digestion. Whereas you can see in the knockouts, you really never see that. You kind of have this steady and this sort of increases. So this is a regional fluorescence analysis on the phagolysosome, on and around the phagolysosome itself. I'm not showing the plot, but the cells themselves, so the regions that aren't involved in engulfment, don't actually change in chloride flux. And I think this is an important point because this suggests that there is a regional chloride flux, and to some extent also sodium flux, which I'm not showing the data for, but there is a regional flux occurring right where the phagolysosome process is happening. And I think that's really important because that suggests that that there might be an effect on the phagolysosome itself during this clearance process. And so again, if you were observing astutely on the microscopy, you might have noticed already some differences. So for instance, if we look again at our control, it brings it in and it pretty quickly disassembles that corpse, right? And so all of those vesicles, the phagolysomal vesicles, are pretty small, despite the fact that he's eating multiple corpses. I mean, it's a really beautiful example of that. Whereas if we look at the knockout, already you can see these phagolysomes are massive. They're really gigantic. despite the fact that they're acidifying, despite the fact that they're maturing and degrading the corpse, it's clear that there's something wrong with the phagolysosome itself. And so Julia quantified this, and lo and behold, they actually have post-engulfment really much larger phagolysosomes that is not observed in the control cells. So what does that actually mean? And I think this is where we took we benefited from work by others who looked at pathogens that infect the phagolysosome and are able to escape. So there's a construct that's able to look at damaged lysosomes, and it does this via a galectin puncta. So basically, the way this works is that you have in the cytosol of the cell, you have lots of these proteins called galectins, galectin 3, 8, and so on, that normally don't have access to any of the lysosomal or phagosomal or endosomal compartments where you have these glycans. And so if you have rupturing or opening of these endosomes or lysosomes or phagosomes, then you have the ability of these galectin proteins to bind the glycans in order to try and actually repair or remove this. But it's a really nice assay because you can essentially see pretty clearly that you get these puncta formed right at where the lysosomes are. And in fact, you get these what are called galectin 3 puncta, and this tells you that there's rupturing occurring and there's damage to the lysosome. And so Julia decided to do this with here. So these are just videos demonstrating this as proof of principle. You see here, this is going to be a super resolution microscopy showing GFP labeled phagocyte. Blue will be your LAMP1, so that'll label your lysosome. Galectin 3 is the indicator, and cipher again is labeling your corpse. So this is what this looks like when you have a phagocyte. Okay, you believe me, this guy ate a cell. Now, if you only look at LAMP1 and galectin 3, you can already see that there are these lysosome puncta forming. And if you just look at the stills, very clearly in these diffused phagolysosomes, you get these really beautiful puncta forming in the lysosomes, suggesting that there are these lysosome rupture events. And if you quantify that, and lo and behold, you actually have quite a significant increase in phagolysosomal disruption, as indicated by galectin 3. So not only do we have change in lysosome size, we also have what appears to be these classic micro ruptures occurring in the lysosomes. And so what's the consequence of that? Well, this is where, when we began this project a bit back, one of the obvious hypotheses was that it had previously been shown that lysosome rupture actually releases cathepsins and can induce the NLRP3 inflammasome, or it alters ion flux, which in turn can also affect the inflammasome. And so for us, that made a lot of sense. And then more recent work had also linked this together with the idea that chloride flux could also be required for the inflammasome assembly. Specifically, the efflux of chloride is necessary for the stable assembly of the NLRP3 inflammasome. And in fact, the paper I mentioned that also suggested Wink1 was necessary for this suggested that Wink1 suppressed this. And so if you lose Wink1, they actually observed more inflammasome activity in response to LPS. Again, suggesting that the lack of Wink1 means that you don't turn on that pathway to bring the chloride back into the cell and presumably shut down this inflammasome. And so that was what we were wondering, was essentially, are we seeing any indicators of inflammasome induction during efferocytosis? So before I show this data, I want to highlight something though. In our field, when we use things like LPS, MSU crystals, and so on, this is a pretty bolus sledgehammer approach. So every cell is stimulated with this. It doesn't take very long to induce transcription and the post-translational modifications that are needed. And you get really quick and really high concentration production of IL-1 and other factors. That's never going to be the case with phagocytosis. First of all, this is a heterogeneous process. In any culture, you will always have macrophages that just don't eat dead cells, and you will have some macrophages that eat a lot of dead cells. And that was even the case in these knockouts. It is always a per cell increase in engulfment. And so I think this is worth highlighting because a lot of what we have to do is by looking by imaging as opposed to sort of conventional Western approaches, for instance. So the first approach Julia took was to look at cleave caspase 1 using the FLICA caspase 1 cleavage assay. So this is a bit of a dirty assay, particularly because the apoptotic cells we use can also label, can also be labeled with the FLICA. So I'm just showing you here that there's really very minimal background in the no AC controls in either the knockout or the controls. You do get some decent amount of signal in the control plus apoptotic cells. But if you actually do the imaging analysis, you can see very clearly that the FLICA is labeling the dead cells, either bound or engulfed dead cells, things we couldn't wash away, or in this case, if it's, if you do, well, this is all fixed imaging. So despite that, there will always be bound cells and it'd be difficult, but you can exclude these via imaging. So if we do that, then there are basically no caspase 1 puncta using this reagent that aren't actively labeling the engulfed or bound dead cells. However, if you look at the knockouts, you see very beautiful puncta. This is very conventional puncta with no overlap of the corpses, bound or engulfed otherwise. And so you can see very, very clearly, and in fact, it's, but even despite the beautiful presence of these cleave caspase, you can see, if we guess that this is 100 to 200 cells in this plane of view, we have 20, maybe 5 to 10% of them have a positive event. And this is after a couple hour period of an experiment, right? So I just want to highlight that this is, while this is a robust event, this is not necessarily what you would see with LPS plus nigiricin, for instance. So we were not entirely thrilled with the background that occurs because of the labeling of the dead cells. So Julia also used an antibody against the ASC. So ASC, if you recall, is one of the proteins that associates with the inflammasome, the NLRP inflammasome complex, and is necessary. Is a necessary component for the downstream signaling, which results in cleavage of procaspase 1 and eventual cleavage of IL-1 and so on. And so ASC is present throughout the cell, but then if you induce the inflammasome, it becomes a puncta. So you get these beautiful specks of ASC. Again, there is no background puncta in either control or knockout cells. And we do not, even though these cells are engulfing, here red, for instance, or white arrows are indicating your engulfed corpse, you don't see these specks. Whereas if you look at the knockouts, you see very beautiful ASC specks and it's usually in the actively engulfing or digesting phagocytes that we see these specks. And so again, maybe 10% of the cells at any given time are doing this, but that is, a meaningful contribution to this process. So both caspase 1 cleavage and ASC spec formation is observed in the SLC12A knockouts that are actively eating. So Julia decided to do something that I think was really cool. So in our field, most macrophages exist in a tissue for a long period of time and often have to, either under the context of homeostasis, where they have to do repeated engulfments over periods of time, right? Daily, weekly, monthly, or in the context of repair. So for instance, in the context of disease models or repair or sterile injury and other sorts of things, where they'll have to eat multiple corpses in multiple rounds. We call this continual efferocytosis. So it's this idea that if you have, you feed one set of corpses and then you can feed a second set. And you can label them with different things in order to see, the second wave of engulfment. And one of the things we've observed is that these SLC12A knockouts also eat significantly more during the second wave of engulfment than the control cells do. So they just never get, satiated. And in fact, this was where Julia saw probably the most robust results, whereas we never saw ASC spec formation in the controls that are undergoing continual efferocytosis. We saw really robust ASC spec in these phagocytes that are engulfing multiple corpses during continual efferocytosis, as indicated by the multiple white arrows. So you can see that they're eating more and that they have this really beautiful spec formation. So I just wanted to, before we get into the last section, remind you of this model now. So this is, we are now working on this idea that there is, that efferocytosis itself, when you engulf a dead cell, primes for an immunogenic response, and that you have to actively suppress that primed response. But if that were true, what is the signal one, right? So classic inflammasome biology tells us that you need two signals. You need signal one that induces the protein production of, let's say, NLRP3 and pro-IL-1, and then you need signal two that really triggers the response, the actual cleaving and activation of that. So if this were true, we would hypothesize that even wild-type macrophages or normal phagocytes would have this prime response. They would have the production of NLRP3, at least early on, and just never cleave it. And so this was, I think, pretty cool, where we could look at a overfed population level. What we can see is really striking is that although there are some basal levels of pro-IL-1 and of NLRP3, if you either feed with apoptotic cells for 4 hours, so if you feed with apoptotic cells, and this is about a 2-hour experiment, or you feed them and then you rest them for 6 or 24 hours, you can see robust production of these, of the NLRP3 and pro-IL-1 beta. This is important. I post this because we can change the biomass, the size of the jerkat. So if we only kill them for 4 hours, it's a whole cell. If we kill them for 24 hours, it becomes apoptotic bodies, so small particulate. And so if we do that, just pay attention to the left side of it. If we feed these smaller bodies, it actually takes longer, but we can still get the production of both pro-IL-1 and NLRP3. And this is important because this suggests that this is a biomass, so amount of biomass dependent phenomenon that's occurring in the phagolysosome. And it's not just a mixed species human mouse, because if we give mouse thymocytes, we still get the exact same response. So there's not some weird cross species effect going on. In fact, it also happens when you feed thymocytes. So I just wanted to take a moment to just reiterate what this means. Essentially, what this means is that normal homeostatic efferocytosis is a signal one, we think, for in the inflammasome induction, right? So instead of giving LPS, in this case, you're seeing what seems to be some production of the inflammasome components. Now, I'm not saying it's as simple as that. There are probably lots of negative or inhibitory mechanisms that are induced during efferocytosis, right? I mean, there's probably lots of redundancies and ways to prevent this potential catastrophic event from occurring. And I just want to add for solidarity that it's not a contamination of the, particularly in the case of NLRP3, but also when we do the thymocyte and get the pro-IL-1, it's not a contamination from the dead cells of you can see here at the 50 kilodalton as well as NLR. It's not just a contamination from the actively engulfed corpses, nor is it detectable MICO or LPS in any of the lines media serum, at least done for these experiments. Because I think those are important notes, right? You don't want to have a contamination that's inducing this inflammasome. That would obviously really disrupt this. Okay, so then this last part, I have to be honest, we've been waylaid, as have probably all of you, in the last year or more, from COVID, so we've done our best to try and do some of this animal work, but it's been stop-start with a lot of disruptions. But at least have some data, some interesting data to show, and make of it what you will. We haven't done a lot of the important histology or IF analyses, but you know, at least we have some indicators that there's some meaningful biology going on in vivo. So what Alyssa first did was just to see if there's any evidence of inappropriate clearance in vivo in our knockout mice. So in this case, we have flux SLC12A2 bred to a constitutive CSF1 receptor CRE, which should target all monocytes and yolk sac derived macrophages in any cell that's expressing the CSF1 receptor. And so what Alyssa did was give bolus apoptotic cells that were labeled, and then basically take blood at 24, 48, and 72 hours, and then harvest tissue for analysis. And so what Alyssa saw was pretty interesting. Basically saw that there was a modest but significant increase in neutrophils that eventually waned after 72 hours. So this basically, only in the knockouts did we see this, you know, kind of influx of neutrophils in response to apoptotic cells. This is consistent with some previous work from Doug Green's lab, looking at the protein Rubicon, which interestingly enough also seems to function during phagolysosomal fusion and for appropriate digestion of apoptotic cells as well. So if they inject apoptotic cells, in those cases, you see this sort of rapid infusion of neutrophils, sort of like a pulse of inflammation. And then I think interestingly, also from this blood, we saw a significant decrease in patrolling or circulating monocytes, which suggests anyway that these cells are either helping out macrophages in the tissues or responding to inflammatory response. And so, but you know, they're basically behaving differently in the context of the knockout. So this already suggested that there was a modest effect in the inflammatory response to apoptotic cell injection, which shouldn't normally happen. And that was consistent with tissue analysis, where we basically saw a significant decrease in the patrolling monocytes, again suggesting egress in response to inflammation. Right, those cells would downregulate CX3CR1, and then egress into the circulation and infiltrate tissues. And I think, you know, this classic apoptotic cell injection experiment, predominantly those cells go to the kidney. So it's not great for testing other tissues, but it's at least we know that those cells predominantly go to the tissues. And what we saw was what you would expect. We saw fewer free dead cells, suggesting that these guys, the knockouts, are eating more. But in that same tissue, we also saw an increase in the total monocyte pool in the kidney as well, suggesting that even though they're doing a better job of clearing the free ACs, there's an opposing, potentially inflammatory response to those dead cells. Now, of course, this is a very acute experiment, so it's can't make any clinical conclusions from that, but I think it is indicative. So then we looked at, you know, what tissues express high levels of the transporter. So all macrophages express it to some extent, but really it was surprising to find that the tissue resident macrophages that are responsible for high burden apoptotic cell clearance. So when we say high burden, we mean really massive, often daily turnover. So red pulp macrophages clear red blood cells, thymic macrophages clear thymocytes, billions of thymocytes. Kupffer cells are also spent red blood cells, but then also cholesterol. And of course, we know microglia do pruning, apoptotic neuron, lipid clearance, all sorts of functions. And so it was really interesting to see that the ones expressing the most amount of this were in fact these high burden tissue resident macrophage populations. So to try and target those, you know, we went back to the constitutive CSF1 receptor CRE mouse, and then started doing aging. Now, the numbers are paltry on this. because we've only been, we had to basically cull our colony prior, during the pandemic. But I think we have some evidence to suggest that there's actually a pretty striking effect, both on monocyte recruitment and changes, as well as and I'll show you some tissue in a second, tissue numbers. But what we saw, particularly after aging them to 20 weeks, and we have more cohorts aging to try and age to a year, which is where Doug Green's lab saw the most significant effect, was really after aging them a period of time, about 50 weeks or so. But what we saw was you basically lose the circulating monocytes over time, and you actually have a significant increase in blood classical monocytes. So your classical inflammatory monocytes, particularly after 10 and 20 weeks. And like I said, with the commensurate decrease in the patrolling monocytes. So total monocytes change because they're probably egressing into tissue, and whatever is remaining, the vast majority of them in the knockout are these classical inflammatory or inflammation-associated monocytes. And then if you look, I'm only going to focus on the microglia right now, or in the brain right now. But we saw similar results in the liver and the kidney, liver and the spleen as well. But what we saw with over aging is we saw these increased neutrophil levels, and there's also an increase here as well, although we need more ends to really see what's going on later on. It's also possible to possibly a consequence of the fact that we know microglia change pretty significantly with aging as well. But at least as far as the monocyte levels as well in the CNS, we see a pretty significant loss of your patrolling monocytes that are really necessary for CNS vasculature function and maintaining homeostasis in and around the CNS. But to us, this really suggested that a lot of the effect was going because of a continual effect on not just the tissue-resident macrophages, but also monocytes that might be compensating for them. So we decided to also breed this to an inducible Cree. So that way the tissue-resident populations develop normally, and then we could try and perturb them using tamoxifen, and then look at how the tissues fared after this pulse of tamoxifen. And so this is all preliminary, but just to show you again, even despite the fact that this is using the inducible Cree, you do still see a similar effect on the patrolling monocyte decrease, with an increase in tissue infiltrating monocytes, both in the spleen and in the brain. And we also saw some modest increase in neutrophils, although this is often difficult to measure in tissues simply because of circulation and such. But it seems like there might be some infiltration occurring, particularly in the brain. And so Alyssa did some really nice imaging just to look at the brain. So this is what a normal control microglial brain would look like. So we just show you. So you can see green is GFAP. This is labeling astrocyte feet, the hind feet. And red here is IVA1. That's really nicely labeling the microglia. And then for fun, I guess, we also labeled caspase 1. This is total caspase 1. You see very little caspase 1 in the control mice. And this is a pristine brain. This is what a brain should look like. Then if you look at the knockout, you can already tell, we call this grandma's test. You can already tell that this is a pretty dramatic difference. And if we look at it, I mean, this is the hallmark definition of both microgliosis and also astrogliosis. And so clearly the knockout brains after tamoxifen treatment were, after deleting SLC12A2, were, there's some element of inflammation occurring in the brain that's inducing these gliosis. And there's also a lot more caspase 1 in the knockout brain, suggesting that there's also inflammatory cell death occurring in those brains. We're hoping to follow up. We just got a hold of the gastromin D antibody, so we're hoping to look at that as well. But if you look, it does also seem to be some cell intrinsic, so microglia intrinsic caspase 1 protein, and we think that this means that these cells are also possibly undergoing caspase-1-mediated pyroptosis, so an inflammatory cell death. And then for solidarity, just so you don't think I'm picking a single region, Alyssa did sort of a multi-region quantification of the potential microgliosis. And in fact, that is the case, that there is a massive increase in IVA1 positive microglia. So this is true microgliosis. So just to sum what I've told you, so this classic model of what we would call the failure to appropriately clear model of efferocytosis, really, this is the hallmark lupus, many lupus patients, atherosclerosis patients. The idea is that you have an apoptotic cell that is either successfully cleared, and then you produce cytokines that keep the neighborhood happy under normal homeostasis, or you fail to clear. So there's floating dead cells. Those floating dead cells become necrotic. In the case of athero, you have the development of the necrotic core and a plaque. In lupus, you get antibodies in response to the necrotic debris, such as double-stranded DNA, and that in turn results in disease. What we're proposing, based off of the data I've shown you, is a new model called the failure to appropriately digest model. And what this suggests instead is that most phagocytes, most macrophages, eat just fine. They can take up corpses. You can do a lot to these guys, and they will still engulf. You can knock out receptors. They'll use other receptors, you name it, and they can do a good job of clearing. What really determines good or bad is how they digest this material. Do they appropriately digest it? Do they appropriately handle the amino acids, the lipids, you name it? And if they do, then that maintains homeostasis. If they don't appropriately do it, like in the case of the SLC12A2 or WINK1 knockout, or that pathway, then this results in disease. And so I think moving forward, this is really what the lab is working on. This was the basis of the Pew that I was very fortunate to be awarded. And I think this was the first indication of this. But if we have a review coming out, I would recommend reading it. We go over many different cases of various histiocytosis diseases, so histiocytoses that involve transporters or other phagolysosomal processes, nucleotide transport, you name it, amino acids, and so on. And so we really lay this out and lay down the case, including in several different human diseases, that all indicate that this might be another way in which autoimmunity and inflammatory disease develop. And I think rethinking terms of human disease, inflammatory disease and autoimmunity, we need to think about how these corpses handle these. And this is particularly true, for instance, in a lot of these lysosomal storage diseases and otherwise. So with that, I will stop and thank the lab and my collaborators and yep, turn it over.
Thanks, Justin. So I'll start you off with the question due to the delay. So do you know about any patient cohorts that have SLC12A2 mutated, and do you know what they present with? And then I can ask another question later, but yeah, I think that's the main one for now.
Yeah, and naturally this is the one you get, right? And there are two patients as part of the NIH Undiagnosed Disease Program. My colleague, who I'm embarrassed to say is not listed, oh no, yeah, he is, Eric Delpire. So Eric is a fantastic scientist out of Vanderbilt, who is one of the leaders in this biology. They've studied these two patients who have an 11 base pair mutation in SLC12A2 that is a hypomorph, so it's a loss of function. So if you lack the protein altogether, I'm pretty sure you don't develop. But this hypomorph, these kids develop kidney disease very, very early on, but we can treat them, and they're just now getting older. And as they've gotten older, we're finding that these kids develop various chronic inflammatory seculi. So early days, but more of this pathway is these patients who originally develop, hypertension or their kidney disease. We can treat that, they can live, but as they get older, we're seeing the manifestations of chronic inflammatory disease in many of them, and there are more and more of these reports coming out. So yeah, I think so long as you get past the sort of early developmental window or the issues with the kidney, then you start seeing what chronic aging, it's aging, right? It's really the chronic inflammation can do to you.
Cool, thank you. So one of our first comments was asking you to just comment on efferocytosis in the circulatory system. So heart tissue, valve tissue, veins, arteries. Just to comment about the process and those.
Sure. Yeah. And I have to give a shout out. So Ira Tavis's lab, his kind of the world expert in this process in athero. And so all of his work is wonderful. And several of his former trainees, if we're promoting trainees, have really branched out into this area. So one of them is Ed, oh my gosh, I'm usually so good with names. And I'm spacing it right now. It'll come to me later, like 30 minutes from now. So anyway, they've been focusing on this in the context of cardiac repair and cardiac damage. And it turns out that there's some really, really beautiful biology as far as the cardiac macrophages necessary for clearance of, for instance, exuded mitochondria. So apparently this is a very common process, is that cardiomyocytes will just dump their aged or spent mitochondria, and those have to be cleared by cardiac macrophages. And then also if you do sort of these damage models, infarction or other sorts of damage models, the tissue resident macrophages are really necessary for cleaning up the debris and regeneration of that tissue. I wish we had that expertise, because I think that's really interesting and cool biology. Hopefully I answered your question, but I would, if I could remember Ed's last name, I would tell you to go read their papers, but I just, can't right now. But it's in Chicago. I even know where their institution is. Anyway, classic.
Okay, we'll move on to the next question. Sure. So
Ed Thorpe. Check out Ed Thorpe's work. Yeah, it's great. Sorry.
We did it. So the next question is if you think that the phagocytes that activate NLRP3 upon inappropriate phagocytosis undergo pyroptosis, and what you think that will modify the chloride flux upon pathogen recognition.
Great. So I actually took this video out. I wish I would have kept it in. So we can observe pyroptosis occurring occasionally in cells. These are difficult because, again, we're not giving a stimulus that every cell receives. We have to be lucky to find the cell that eats and undergoes pyroptosis, right? So if you have 100 cells and only 10 of them eat, let's say, or 20 of them eat, and only a fraction of those undergo, let's say pyroptosis, you have to get lucky enough to capture it during the eating and follow it during the pyroptosis. Now, having said that, we have observed this. It's just not easy to quantify on a population level. Having said that, I think this is actually leads to, for instance, it is already known. So bumetanide is the drug that targets this transporter. Millions of people are given bumetanide for hypertension. And so there have been some really nice studies. There was a really nice study that actually showed that if you give bumetanide during Klebsiella pneumonia infection in mice, that you have significantly better outcome in response to Kleb pneumonia. And in fact, there's another paper that's a bioarchive that also shows similar results in microglia with regional bumetanide versus circulating bumetanide, and then using this drug targeting this transporter as well. So I think I answered the second part of that, but essentially, yeah, I think targeting this pathway, this would be really cool to do. Our neighbor is a TB expert, and we've been trying to get this set up, but hands and time. But I think there could be a really nice way to target these sorts of phagolysomal pathogens to try and better, attack or kill those pathogens.
So last question seems that they want your comments on specific signaling from inappropriately digested PAMPs from phagocytosed microbes and how that regulates your chloride influx or efflux.
So I have no data on that, nor do I have any data how, for instance, endolysosomal or these sorts of TLR signaling inside of there, how that works. We're going to try to do some of this. Like I mentioned, the Wink-1 knockout paper looked at, classic, LPS induction, extracellular LPS. I think we're going to try and do some of this chloride imaging with LPS and with some of these other, because my guess is that LPS, we know, at least when you give extracellular LPS, induces a macropinocytotic or an endocytic event. And so I would expect, similar to what we see for macropinocytosis and what we see for efferocytosis, I would expect these regional fluxes. I think the reason why it might be difficult to tease that out just experimentally is because when you give a PAMP in any way, it's not just like one event, it's the whole cell often. There are many receptors signaling at many different places. So we'll do the experiment. I'm curious to see what it does, but I could see it not working simply because it's really hard to tease out regionality when you have many events occurring at one time on a cell. Whereas with apoptotic cell, right, like it's, one here, one here, one here, so you can tease that out. So my hypothesis is that you will see the similar phenomenon. I'm just not confident that we'll be able to experimentally tease it out. But like I said, we'll do it. That's a great question, though.
Okay. Thank you, Justin. Thanks, everyone, for joining us today. Our next talk will be next month, and we hope to see you all then again. Thanks for stopping by, and have a great day.